Welcome back, friends. In this video tutorial, we'll be talking about transgenes and transgenic mice and how we produce transgenic mice. So, what is transgene actually? Transgene means simply we have a, let's say we have a particular gene of our interest which codes for a particular protein, the expression of which is known for us, sometimes unknown for us. We want to see the expression of a particular gene in a living organism, for example in the living mice. In that case, we need to inject that gene inside another mice to see the effect, right? So as we take our desired gene, let's say this is the gene of interest with this red color in middle, and we have other constructs. So because it is a vector, which is a gene delivery system, so we'll take that gene and insert it inside a mouse. That is called the transgenic mice production, because once we take our gene from outside, inside a vector or the gene delivery system, and we deliver it inside uh, a in vivo system like a organism like mouse or rabbit, in that case that thing is called as a transgenic system, and that's called the transgenic mice or transgenic rabbit, whatever we produce. So in this case, this mouse over here will be called as the transgenic mouse because we transferred a gene from outside inside this mouse. So how can we do that? For the general process of this, we need to have an embryo, because the embryo which is fertilized oocyte, for example here, it carries all the potential to develop into a mouse, right? So what we do, we actually take the gene of our interest, which is here in this one, and we inject this gene, which is obviously incorporated in vectors. We take the vector with the gene of interest and inject it inside the fertilized oocyte, which is denoted here with this round. This is the oocyte, and we take the injection, that's called the micro injection, and we inject the gene of interest inside that oocyte. So once we do that, after that what will happens, it will transfer that mouse. So we take that oocyte containing the gene of interest carrying vector, and then we place it into another mice, that's called the recipient mother mouse. In this case, generally it's a foster mother, so it's called the recipient mother. So once inserting it into the recipient mother, we look for the progeny, and the progeny, among the progeny, some of the mouse are going to get that gene, and those will be called as the transgenic mice. Some of them are not going to get that gene, and they will be called as non-transgenic mice. So this is the overview of how we produce transgenic mouse. So now let's look at the much more detailed stages over here. So if you look at here in this particular case, we can actually divide the production of transgenic mice in two different sections. One is called as the initial phase and the insertion of gene of interest inside the embryo, and the second phase is called as the development of the transgenic mouse. So if you look at here, this is the left hand side, this is the first section, and in this one is the incorporation of the gene, the transgene. Transgene means the gene which is incorporating from outside inside the organism. So what we need to do in this case, we need to produce a vector. That vector will contain the gene of our interest. For example, here if you look at here, this is a vector. It can be a plasmid, or it can be, if the gene of interest is larger, we need to take vectors like yeast artificial chromosome or yak, or bacterial artificial chromosome. So we take it, let's say the yeast artificial chromosome, and we insert the gene of interest, in this case denoted with this red colored segment, into that vector. We clone it. That's called the molecular cloning. That is the very first stage. Once the cloning is done, we have the gene incorporated to the vector, which is the delivery system, because we cannot directly insert this gene. We can insert this gene directly also, but this is one of the process of doing it. Now once this is done, then what we do, we need to also take the fertilized oocyte. Now for that, what we need, we need to have the embryo donor, and we not only need the female, we need the male also, because we should carry that fertilization process there between this embryo donor parent mice. As you can see here, these are the parent mice of embryo donor, and after the fertilization between them, what we get the embryo. In that embryo, there are different sections of the embryo, and one is called the inner cell mass, another one is the outer cell mass. So what we look for here is the inner cell mass, because the inner cell mass contains a lot of cells which are called embryonic stem cells, or ES cells. They are very very important, and these cells are totipotent in nature. That means these cells can give rise to different, all the different type of cells of the body. Like these embryonic stem cells can produce nerve cells. These embryonic stem cells can also produce liver cells. So they are very much versatile here. So we take some of these inner mass cells and put it into the cell culture. Why? Because we want a lot of same type of cells during the cell culture. And during the cell culture process, we select certain cells. We produce the cell line with a particular type of embryonic stem cells. So once we get that cell line in our hand, we take that ES, or in the embryonic stem cells, out of that plate, of the cell culture plate, and then we inject this vector, the clone vector, inside that embryonic stem cell. Now this injection or delivery of our desired gene inside the embryonic stem cell may occur in three different possibilities, and we are going to see those three possibilities here in a little later. But in this case, this is very very important. So let's look at here how this process actually work here. So this is how the DNA is injected there. So if you look at here, this is, let's say, the gene of our interest. Let's say this is the gene of our interest in these curvy lines, and we have the vector which is called YAC, or yeast artificial chromosome. Now what we do actually, we insert this inside a yeast cell, so we have a yeast clone. Now what are the ways that we need to inject this DNA of interest inside the embryonic stem cells? So one way is the spheroplast fusion. In the spheroplast fusion, what happens, we have this embryonic stem cells which is here brown in color, and we fuse it with the yeast clone so that those gene is inserted inside the ES. Second way is the liposuction or simple micropore injection, or not injection actually. In this case, it can be liposuction or the electric current that is provided to the cell membrane of embryonic stem cells, which changes the voltage and also creates pore in the cell membrane. Creates pore by separating all those phospholipids, so that it creates a channel. Through that channel, that gene of interest can insert inside the ES. Now the third way is the micro injection system. The micro injection system is, you have also the cell, the fertilized oocyte, and inside the fertilized oocyte we just inject the desired gene there, the desired gene with the vector. In that case, we don't need the complete yeast clone. Instead, we just require the purified yeast artificial chromosome. But remember, in spheroplast fusion, we need the ES cell as well as the yeast clone, because we need to fuse two types of cells together. They require this cellular cell membrane to have fusion. But other two processes like liposuction, microporation, or microinjection techniques, we only need the vector containing the gene of interest, and we inject it into the fertilized oocyte here, and then we implant it into the cell, into the organism. So if we go back here, so once that process is done here, after that the second phase starts, and the second phase is to deliver that cloned cell, that deliver the embryonic stem cells inside another embryo, and then implant that embryo to the carrier or recipient organism, or the recipient mouse, to develop into the proper progeny mouse containing the desired gene of interest. So here we have the embryo host parent again, and they produce this fertilized kind of embryo. So once we have that embryo, then what we do, after getting all this cloned vector inside the embryonic cell, we again go for several rounds of cell division in cell culture lab. So after that, among different cells, as you can see, there will be some cells which are the recipient of the vector, and also there are some cells which won't get any vector containing the recipient at all. So in that case, we need to select only the cells containing the desired gene with the vector. So let's say these are the cells. So as you can see here, these are the cells. So we take that cells and then put it inside the, again, the inner cell mass area of the new embryonic stem cells of the host embryo. So we inject it there. So once we inject that, Because why we do all this cell culturing thing? The idea here is to produce the nature, that all the cells that are present there should have the proper nature. All the cells that will be placed there, they have the potential to grow and develop, and they all have the desired gene with the clone vector inside. So that is very very important. So once that thing is done, then we inject it inside the embryonic stem cell, inside the inner mass area of the embryonic stem cell. And then what we do, as you can see here, the new embryonic stem cells are designated with the red coloration, right? So after that, the stage will be like this. So we have this mouse now. This is the recipient mouse. So we need to inject all those embryos. Remember, these red things are not only means the embryonic stem cells, but instead we take all the embryo and we inject those embryos inside the uterus of the recipient mouse. Once we inject that, they will provide the offspring, the progeny. And if you look at the progeny here, they are chimeric in nature. That means in this case, though this mouse is white in color, but we take it from the brown mouse in that case, for example. So all of them kind of have some of the cells of the progeny mouse's body contain the transgene, some of the cells are not containing the transgene. But what we require here, we need to produce transgenic mice which are completely transgenic. That means which are transgenic for homologous. So homologous transgenic mice, homogeneous, I mean transgenic mice. Not, it should not be the heterozygous, it should be homozygous uh, transgenic mice, right? Sorry, I told homologous, actually it's homozygous transgenic mice. We all know what's homozygous, what heterozygous is. So once we have this progeny mice, we take one of this progeny mice and we cross it with another mice from the system, which is a wild type from the system, to find out exactly whether our embryo is implanted or not. Now after this cross, what we can have one of the heterozygous mice produced. Now the heterozygous mice, as you can see here, is in brown in color here. So we got this heterozygous mice here. So if I take an ink here, so as you can see here, that due to the cross between these two, it produces this heterozygous mice. So once we produce this heterozygous mice, we cross this heterozygous mouse with another heterozygous mouse, and again this is another simple cross. So another cross will take place, and then after this cross between two heterozygous mouse, what we can expect? Now using the common idea of Mendelian genetics, we know if two heterozygous organisms are crossed, there is a possibility of developing a homozygous, right? So in that case, what we do actually, we generate three different possibilities. One is the possibility of producing homozygous wild type mouse, and that the possibility is 25%. Second possibility is homozygous transgenic mouse, and also the possibility is 25%. And the maximum chances of producing is the heterozygous transgenic mouse, that is a 50%. So if you look at here, what we require among all these three different types of mouse, we require the homozygous transgenic mouse, because this homozygous transgenic mouse contains all the desired, all the different genes, I mean all our desired gene in different cells of inside their body. So that's why it's very very important to only get this homozygous transgenic mice, and the percent is 25% only. So that's how we produce the transgenic mice. Now, if we look at the genetics of how this transgene is incorporated inside the vector, if we look at here, this will be very clear that this is the vector for interest. Let's say this is the vector, and this vector, as you can see, it contains a particular allele. Let's say the allele is the A star here, and it also contains a neomycin resistance gene, and also different other sections of recombination sites here, the which is denoted here in this blue color section, which are also called as regions or regions of homology, because they have the homologous section between them. So the idea here is we also have the original allele, that is A in red color. So there will be exchange of the genetic segment between them. So there will be two different types of producing the transgene here. One is that if there is a recombination take place between this vector and this desired gene of interest here, so due to the homologous recombination between these blue sections or blue segment of the genome, this A star will be replacing the A, right? So the A is the actual original allele we need to replace. For example, it will be replaced with this A star, right? So if you need to do a knockout, this could be done. This is the process of gene knockout actually, where we actually block or delete the activity of a particular gene with the help of insertion with another gene. Now if we look at the insertion, actually we can also take care of this random insertion. So where we have the gene of interest here, I mean the vector and our desired gene, and we insert the gene for the vector, that is neomycin resistance, that TK or A star genes inserted inside the desired gene. So in both the way they can be inserted, and what that can do, they can actually change the different properties of that organism, right? So for example, if the strain after this random gene insertion, it can be killed by ganciclovir, which is a drug. On the other hand, if the first thing happens, like the replacement is happen in this first case, that particular cell will be resistant to both ganciclovir as well as G418. So this is a kind of example of how this process works, and I hope that's helpful. If you like the video, please subscribe, share the video, hit the like button, and thank you.